So good morning and welcome to everybody. My name is Davide Vetrano and I am one of the organizers of this symposium. It's a great pleasure for me to introduce the next keynote speaker, this time live, Luigi Ferrucci. Luigi Ferrucci is a geriatrician, epidemiologist, and a very well-known researcher in the field of aging. He's the current scientific director of the National Institute of Aging in the US. And his talk today will be about biology of aging and multimorbidity. So, Luigi, please.
Thank you very much.
The floor is yours.
So thank you very much to the organizer and to Laura in particular for inviting me for this very interesting meeting. Certainly, this is a very, very hot topic in the area of geriatric and gerontology. I will try to give you a perspective that is more from the biology of aging and in the area of geroscience. What I think are the roots, the biological root of multimorbidity, and how hopefully we could use this emerging information on the biology of aging to change our approach to multimorbidity in the future and to create what I hope will be the new class of geriatrician. Okay, so this is a trajectory, an imaginary trajectory of what the aging process looked like. And in a way, we start and we improve our ability to be mobile very, very quickly over the lifetime. And in very few years, we start running. And so, but this program, this first period of life, it's very, very tight. It's very, very strict. It's coded by a genetic program that is encoded in our DNA. And in fact, if you have a child and your child doesn't walk at the 1 year of age, you go crazy. You go to the doctor, you try to understand what's wrong with that child, because it's so much a template of what's going to happen that you expect certain steps to occur. If they don't, you know there is something wrong in the biology. But then you have a period of relative stability, and this period of relative stability, if you don't want to run a marathon, you can pretty much do whatever you want and enjoy all the opportunities of life. And at some point, you have a decline of physical function. And unless you have a sudden death, you go through a period of disability. Now, we have been concentrating a lot in this period of life. And when we think about geriatric and gerontology, that's what we think about. The classic geriatric patient that comes to the observation with multimorbidity and emerging disability or impairment complicated with social and psychological problems. But in fact, what's happening here is actually have a root in things that are happening during this period of time. And I will try to give you an idea on how and why what's happening in this earlier period of time is so important and determining the trajectory of health and physical function that occur late in life. And I think that we are starting to accumulate knowledge that we will translate into early intervention in the years to come. What I'm going to do today is limit my discussion to humans. There is a lot that's been done in the biology of aging in animal model. We never know whether these animal models are translatable to human. And, whatever my scientists tell me, there is a lot of difference between a mouse and a man. And I think that's really important because many of the things that we learn from animal model tell us about the biology, but when we try to translate them in humans, they really don't work. And so what I'm going to tell you today, what we know about humans and some of the recent implementation on this measure in the Baltimore longitudinal study of aging and in other studies. This is a slide that you probably know very well. there is an increase in the prevalence of many chronic disease. I don't have to tell you to this audience. Even for the disease where you see a decline, such as hyperlipidemia, for example, or diabetes, probably you're dealing with selective mortality or with frailty, where you have an inversion of the direction of risk factor. This is not because we become healthier in older age. And so it's because of this really incredible impact of aging as a risk factor for many chronic diseases that are at least apparently pathophysiologically unrelated. We think that there is an emergence with age on the global susceptibility that make our failing in the homeostatic control and lead to the emergence of multiple disease. And in fact, if you think about that, if you look at a trend of number of disease, again, I don't have to tell you in this audience, you see that there is an increment of the number of disease by age. What's really less known is that if you look at longitudinal cohort, and each one of these represents a longitudinal cohort in the Chianti study, but data from the Baltimore longitudinal study of aging are the same, you see that there is a geometric acceleration in the emergence of multimorbidity with aging. And so what is causing that? What is behind this general susceptibility? Just fourth sentence. 80% of medical beneficiaries in the United States have more than 2 chronic diseases. Multimorbidity is the most prevalent chronic disease. That is, to me, is unbelievable. Because if you look at the guidelines sometimes talk about comorbidity for disease that have the same pathophysiological risk factor, but they don't talk about how to address disease that are, co-present, but certainly change the prognosis, the clinical evolution, the response to treatment. And so you are in front of clinical picture that are very, very difficult to address according to the standard medicine that you have learned while you became a doctor. And then I wanted to address this with the geroscience paradigm that say that there are some biological mechanisms that determine this global susceptibility. To do that, I need to walk you into a metric classification. And this metric classification here is the way I think about aging. I think about aging as having 3 metrics that are hierarchically organized. The first one is this metric of biological aging, and the last one is functional aging. Of course, what we want to do is maximize this. Our goal is to say people need to be able to walk, need to be able to be independent, need to be able to think, need to be able to remember, needs to be able to not be sad or depressed, not have a problem in mental disease. But in order to do that, we know and we'll learn very much that we need to understand the phenotypes of aging. And so the symptoms, the multimorbidity is what we are really interested. But also, in order to understand the root of this, we need to understand what is wrong and what is not working at the molecular level. So there is a hierarchical organization. And what I'm going to talk to you about is this part. What are the biological mechanisms that occur that eventually, through a number of failing the compensatory strategy and resiliency, lead to functional decline? And I'm going to organize my talk with the, starting from a famous manuscript that was published recently by Carlo Lopez-Otin. This is probably the slide that is most shown in any meeting on aging. But I'm going to try to limit this to what is actually measurable. Many of these are very beautiful. This paper, I probably read it 10 times. Every time I read it, I get more mad because there's no mention of humans. Everything is done there in mice and in rats. And we don't know. And at that time, I didn't really know whether this measure could be done in humans or not. And some of them actually can be done. And so I'm going to limit this to some of them that I think have been starting to measure the human, show you some data. First one is genomic instability. There are entire laboratories and organizations that work on DNA damage and DNA repair at NCI, at the NIA, But if I tell you that there is no 1 iota of evidence that in human there is accumulation of somatic mutation with aging, that is absolutely true. And when we realized that, in collaboration with some of my friend at Albert Einstein, in particular Jan Big, we decided that we needed to study that. We needed to see whether this accumulation really exists. And the way to do it, this is the way we use. Now we're using a method that is a little bit less time-consuming and expensive, but we took CD4 lymphocytes, and then we isolated a single cell from CD4 lymphocyte, and then we took a group of cells, and then we took 3 cells in this study, 5 cells in this study, and we amplified the DNA from single cells so that we could sequence exactly what is the sequence of nucleotide in each one of these cells. And then we sequence also the DNA extracted for a group of cells. Now, because the somatic mutation occur at random in different cells, when you sequence a group of cells, those single defects are not evident. And so you have what is then DNA of that individual, and they compare the DNA of that individual, this template, to the DNA of each single cell. And then you can count the somatic mutation. And when you do that, you find that, in fact, the number of somatic mutations increase with aging. It's happening because some of those somatic mutations are repaired. And because it's not even linear, but it's geometrically increment, that's probably has functionally important. We are validating this for a much larger population, but it's kind of an expensive project because imagine that you need to do whole wide genome sequencing in each single cell, and so you can't really study thousands of individuals. Hopefully, this technology will become more affordable in the future. One that has received a lot of attention that we are working with in collaboration with Steve Horvath is the epigenetic changes. And there are many, many different types of epigenetic changes, especially histone acetylation in this study, but it's very difficult to do in large population in humans. But one that everybody is working on, and it's really showing incredible result, is methylation. And what is methylation? It's simply the transfer of the methyl group to a cytosine that is juxtaposed to the guanosine, so we call that CpG methylation. That's why we call it CpG, is cytosine, a phosphate, a phosphorus, and a cytosine, guanosines juxtaposed, and then you have these are the ornament here that are on the cytosine. And these are important because change the gene expression. We believe that they are mostly in the middle of the binding site of transcription factors, and so that they regulate the gene expression in several sites. And why this is so important? Well, I remember when I came to NIH for the first time, it was 1985, and I had a discussion with Andrea Lacroix. She told me that what we really don't have is a biological biomarker of aging. We are off to rely on chronological age, and this is not good enough. Well, today, if I tell you that if you have a drop of blood that is in the corridor, and you analyze that drop of blood, and you can tell exactly the age of that individual that lost the blood. I couldn't believe that 10 years ago, but nowadays, I'll tell you that's true, and your error will be 1.5 plus or minus 1.5 years. This is a fundamental change in the way we look at aging, because we now know that it's not just the stochastic accumulation of defect. There is some biological process that is reproducible, that is occurring in every tissue in our body. It's harmonic in every tissue of our body. And in fact, I'll show you some data that to me are absolutely amazing. This is the original paper from Steve Horvath, and showing that in a training set, in different tissues, there is an accumulation of DNA methylation sites, and then when you apply this metric that is constructed by 353 weighted CpG sites, you can pretty much replicate the same. And from that original paper, this has been done in dozens of different data sets showing exactly the same distribution. And you can measure the muscle, you can measure the brain, you can measure the blood, you can measure the skin, and you find exactly the same distribution of methylation. Now, not only that, but if you look at gestational age in material that was extracted by fetuses, you can find that the methylation also track, gestational age. And if you take a retina that was taken by fetus that were aborted, you can find that the methylation age in the retina is also predict gestational age. That tells you that although we think that there is something that is so powerful that apply a genetic program early in life, you can tell that aging is already occurring. Some biological aging is already occurring. These are longitudinal data. I mean, these have never been published before. And so if you have ever worked with longitudinal data, you know that longitudinal data go up and down, and then you create some line that you think is the trend for that individual. This is the first time in my life that I see that each single person here was studied 3 times, and you can see that the longitudinal data of the epigenetic clock, it's unbelievably, rely on aging. And what is interesting that when you look at this epigenetic clock, there is an increased variance that occur with aging, as you expect, as I hypothesized at the beginning. Well, the problem with the epigenetic clock that was originally created by Steve Horvath is that we use chronological age for tuning it. And of course, you can tell about chronological age by looking at your watch, and to the second, you know exactly by looking at the driver's license. So you don't need that. So you really need to tone the epigenetic clock by the health and the accelerated aging of an individual. And so what we did was create a second-generation epigenetic clock that was tuned on biological aging. I can talk to you about how we expect biological aging. When you do that, then the effect on different outcomes, such as mortality and comorbidity, is incredibly strong. It's become really, really a strong predictor of health. And what is also interesting is that this new epigenetic clock it's changed according to what you expect. If you look at risk factors, you can see that is lower in individuals that have a certain diet, have high socioeconomic status, with moderate alcohol consumption, with exercise, and is accelerated in individuals that have inflammation, high systolic blood pressure, high triglycerides, and high cholesterol. And so it's not only, it's really the interface between the genetic and the environment, so it can change over the lifetime. Telomere length, as field, probably, and there are Nobel Prize, and there are a field of many of the newspaper, but in reality, it really doesn't work very well. If you look at longitudinal effect on telomeres, you see that they're very, very different. And there's up and down that really doesn't allow you to do a prediction. And so if you look at the shape of the slope, most of the individuals actually have an average increase in telomere instead of a decline. I will skip this. I want to give you some idea. I'm going to skip proteostasis because I want to talk to you about what is the most promising area of research in this field, which is cell senescence. What is cell senescence? Cells that are endangered by gDNA, the genomic instability, and a number of other metabolic stress go through a pre-program change that's called cellular senescence. Cellular senescence is characterized by the fact that they become larger, they have specific change in the nucleus, aggregated chromatin, they express an antigen that's called P1641K. And then they also start secreting a number of substances that are inflammatory cytokines, growth factor, matrix proteins, and other proteins. And they stop replicating. Because they stop replicating, they accumulate in tissues, and the accumulation in tissue makes the tissue no longer functional. And also those cytokines are determining a number of problems that probably are responsible of degenerative disease. Norman Sharpless looked at the p1641K in lymphocytes and found that, in fact, the number of senescent cells increases with aging. Not only that, but the number of senescent cells will predict who has side effect of chemotherapy or was not side effect on chemotherapy. And also, recently, senescent cells were accumulated into intramuscular fat in an American group showing that those who had more senescent cells in the intramuscular fat tend to have reduced mobility, lower muscle strength, and also central obesity. And finally, in the Leiden study, the number of senescent cells in the skin was strongly associated with wrinkling, and also when people judged about the apparent age, those with more senescent cells looked older compared to those with less senescent cells. Recently, we had done a proteomic study in discovery proteomic, and we had measured all the senescent-associated secretory phenotype protein. You see that with aging, there is strong enrichment of this protein in the circulation, suggesting that a biological index can be created that from the blood make you estimate the burden of senescent cells. Mitochondrial dysfunction, I'll only take to that, and then I'll get to the end because my time is going, but this is really another part where we know that this is absolutely important. How do we measure in humans mitochondrial function? We measure by the fact that the muscle and the brain work energetically in a very specific way. They cannot rely on mitochondria directly, because they need to have access to energy promptly and anaerobically. And so they accumulate energy into a molecule that calls phosphocreatinine. We use that to measure phosphocreatinine with MRI. And so we exercise people in the magnet, and when they exercise, the phosphocreatinine goes down, and then we allow them to recover. And we know that the mitochondria replace the energy in the phosphocreatinine, and how fast the mitochondria are replacing the energy in the phosphocreatinine tell you how much the mitochondria is working. And so you can calculate what's called kPCR, that you know change with aging tremendously, although with this large variability between individuals. If you look at kPCR, it's strongly and independently associated with walking speed in many different tasks, mostly in the longest task. And then if you do a mediation analysis, you'll find that the effect of mitochondrial function on walking speed is explained in large part by the effect of muscle strength. So the decline of mitochondria is really what is affecting those. And I will skip this because I don't have the time. I will just go to my... So I show you some examples, not all of them, that these parameters tend to accumulate with aging humans. We remain to see whether they affect multimorbidity or disability or not. We have not accumulated enough evidence to do this analysis. But what we know is that each one of these accumulations is a cause of inflammation. And because the inflammation is really the most powerful predictor of multimorbidity, not only cross-section, but also longitudinally that we know about. Not only those with inflammation and more multimorbidity, but also those with increasing inflammation develop more multimorbidity with aging, that we can assume that inflammation is just the external parameter that read the burden accumulation of these hallmarks of aging. And disability is also strongly predicted by inflammation. 2 last slides. So I told you that here is chronological age. In chronological age, it's not very useful, but it's certainly correlated with aging. But it's a linear concept. At each time, the increment of 1 day represents the same change. But we know that with aging, that's not true. What's happening with aging is that there are profound change initially, less change in the middle, and a lot more change later on. But the variance of these changes increased tremendously with aging. And we have seen that, for example, in the epigenetic clock. Why is this important? Because in fact, what's going on is that there are a number of entropic forces that are challenging our biology day by day. But evolutionary, we have selected a number of compensations that take at a hold the fort against these entropic forces. The problem is that these compensatory strategies are fading over the aging process, and that this is reducing our homeostatic capacity and putting us in the area of accelerated aging. And in order to solve this problem, we only have addressed damage, but we have not really addressed the mechanism of compensation. We can't really avoid addressing those. We can't understand what the different compensations are. We will not address this problem. There is one last issue. Biological aging probably failed early in life here, but doesn't really translate into phenotypic aging because there are so many biological compensations. You can fold the protein, but it can be refolded. You can damage the DNA, but DNA get repaired. At some point, you will have an effect on phenotypic aging, but for example, even a reduction of muscle strength as much as 40% will not impact our ability to walk, because you compensate it with other strategies. And only when you're no longer compensating, you will have the area of biological aging that is affected. That's really important because we are acting here at the time when all the compensation in the body are gone. And we can do a miracle. We can do incredible things. Unless we can act on compensation at the time when compensation it is possible, our result will always be disappointing in the end. And of course, I'm blathering, but these are the people that do all the work that I'm showing you, and I'm so lucky to be working with them. Thank you so much for your attention.